I'd like to thank the DDL Organizing Committee for inviting me to present my work on evaluating the transport and intracellular uptake of drugs across nasal epithelial cells and in neuroblastoma cells. So thanks to Fabio's wonderful and fantastic presentation and overview, I'll just be going through a brief background, specifically of interest in my study. So within the brain targeting aspect of nasal drug delivery, I'm looking into the olfactory pathway and the olfactory region. So within this region, we've got the olfactory nerve cells that connects the nose to the brain through the connection between this nerve to the olfactory bulb of the brain. This makes this region uniquely permeable due to the neuronal pathway. There are some benefits, including the fact that it bypasses the blood-brain barrier, which is a major hurdle for drug delivery to the brain. It's fast-acting with quick onset and is a non-invasive method of drug delivery, which can improve patient compliance. However, we've got challenges, such as the fact that there is a highly resistant and narrow vestibular region before we reach the olfactory region. The region itself is also located in the apex of the nose and has a small surface area. There's also biological barriers, such as the semipermeable membrane and enzymatic degradation. So when we're looking at the in vitro nasal drug delivery model, it is an important aspect of the intranasal drug formulation development. And the current model that's widely used is the utilization of human nasal epithelial cell line, RPMI 2650, that's cultured under air liquid interface, as shown in the diagram on the right there, which allows for the cell differentiation for mucus production. However, there are limitations to this model is that it's not fully representative of the nasal olfactory region, as it lacks the sensory neuron-like cell that contributes to the nose-to-brain interface. So creating a model with inclusion of neuronal cells can create potentially an in vitro model that's more representative of the nasal olfactory region. So the aim of my study is to lay the groundwork for a co-culture model that better represents the nasal olfactory region. And to do this, the first objective, I looked into the evaluation of in vitro permeability of 3 model drugs, ibuprofen, levodopa, and sertraline, at 2 concentrations using the current in vitro model with RPMI 2650 cell. So these drugs were of interest as they are brain targeting and they also have some differences in molecular size, log P values, and charge. So the second objective of my study was to assess the drug uptake of a neuroblastoma cell line, BE2M17, using indirect co-culture model to explore correlations between the drug intracellular uptake and the lipophilicity. So the reason this neuroblastoma cell line was chosen, it's a human cell line. It is adherent in nature, and it also has the ability to differentiate to neurons with treatment of retinoic acid. So for the method, we used the nasal epithelium air liquid interface. The cells were cultured in SnapWell on the air liquid interface, and we performed transepithelial electrical resistance measurement, as well as drug transport and uptake, where drugs are applied to the cells in the SnapWell, and samples were quantified using high-performance liquid chromatography. Looking at the results for the transepithelial electrical resistance and the measurement of epithelial integrity, these are the results for the 3 drugs, the model drugs that we tested. We didn't see any statistically significant difference in the TER values between before drug treatment and after drug treatment, which shows no detrimental effect to the nasal epithelial cells. So this absence of significant TER reductions does confirm barrier integrity during the transport assays that were performed. So for the drug transport, we've got 2 graphs here. So we've got ibuprofen represented by the red line, levodopa represented by the green line, and sertraline represented by the blue lines. So it shows the percentage of drug transported over time. So for ibuprofen, when we look at the red lines, it shows a consistent drug rate transported across both concentrations. When we look at levodopa, it showed an initial slow transport, but it does have a significant increase from 60-minute time point onwards. For sertraline, it also showed an initial slow transport, but it does show a marked increase in drug transport rate at 30-minute time points onward. When we look at the 3 results comparatively, we can see that out of the 3 drugs, ibuprofen showed the highest rate of drug transported, followed by sertraline, and lastly levodopa. And when we look at the second objective of my study, which is the drug uptake, we use indirect co-culture model So in the diagram here, we've got the combined model of both the nasal epithelial cell, RPMI, together with the neuroblastoma cell, BE2M17. So the neuroblastoma cells were seated at the bottom of the basolateral chamber to look into the drug uptake. So the drug distribution result graphs here, we've got the 3 drugs. And when we look at the transported drug distribution, so that's shown by the blue bars there, you can see that ibuprofen had the highest amount, followed by sertraline and lastly levodopa, which does match with the drug transport result. And looking at the intracellular drug uptake of the nasal epithelial cell RPMI, that's shown in the green bars, we've got the highest amount for sertraline, followed by ibuprofen, and lastly levodopa. And when we look at the intracellular drug uptake of the neuroblastoma cell, it showed the same pattern. So we've got sertraline, and then following that, we've got ibuprofen, which is a lot smaller. Unfortunately, you can't really see it in here in the yellow bars. And it was found to be undetectable for levodopa in this case. And when we look at the drug distribution that's found on top of the cells, that's represented by the red bars. We've got levodopa having the highest amount. The second is ibuprofen, and lastly, it's sertraline. So looking at the differences in drug transport and uptake result, one of the factors that we looked into was the log partition coefficient, which provides an indication on whether a drug is more likely to be absorbed by living tissues. So if we've got a high log p-value, they're more hydrophobic, and if we've got a low log p-value, they're more hydrophilic. So according to Lipinski's rule of 5, the ideal log p-value for good drug absorption is between 0 to 5, with an ideal range of between 0 to 3. And when I look at the log p-value of the 3 model drugs that were tested, we've got ibuprofen with a log p-value of 3.5, which falls within the good range and just outside of the ideal range. We've got sertraline, highly hydrophobic, at log p-value of 5.1, which falls just outside of this range. And lastly, we've got levodopa at negative log p of 2.39, which shows that it's highly hydrophilic, but it also falls the furthest away from this range. So looking at the drug transport result with ibuprofen having the highest rate, followed by sertraline and levodopa, it shows that it correlates well with the respective log p-value on how well it is expected to be absorbed by the living tissue. And when we look at the drug uptake result, sertraline having the highest, followed by ibuprofen and levodopa, it does correlate well with the lipophilicity of the drug, with the highest lipophilic drug, sertraline, having the highest intracellular uptake. In addition, levodopa is also hydrophilic and may remain on the apical chamber. And when we look at the previous result with the drug distribution, we do found levodopa to remain at the highest amount of levodopa found on top of the cells. And sertraline, because of its lipophilic nature, it does rapidly partition into the membrane and accumulates intracellularly, which is shown here in the drug uptake result. And in summary, there was no detrimental effect by drug observed to the nasal epithelial cell through the measurement of the tight junctions. The drug transport result showed ibuprofen to have the highest transport rate, followed by sertraline and levodopa, which correlates well with the respective log p-values. In terms of drug uptake, sertraline exhibit the strongest intracellular uptake and accumulation in both cell lines that were tested, so the nasal epithelial RPMI and neuroblastoma BE2M17, which is consistent with its high lipophilicity and its ability to partition into cellular membrane. So in case of this study, log P shows a bigger role in drug transport and uptake compared to other factors such as charge. And this results from the drug transport and uptake does show that this preliminary co-culture model is physiologically relevant, and we're looking into future studies into creating and developing a more complex co-culture model in air-liquid interface. In the diagram at the bottom there, so we've got, the plan is to seed the neuroblast cell, which would be differentiated into neurons on the underside of the apical, so it creates a more direct model of co-culture. And last but not least, I'd like to acknowledge and thank my supervisors, Dr. Ong, Professor Trini, and Dr. Suman, the Respiratory Technology Group team members, the Woolcock Institute of Medical Research, Apta Pharmaceuticals for the scholarship provided to me, Macquarie Medical School, and Macquarie University Graduate Research Team. Thank you.
Thank you very much for that lovely talk. Are there any questions? There's one over there, you can see.
Great talk, thank you. So the co-culture model is of course very interesting, and you showed that the lipophilic component was most actively taken up by your neuron cells. However, they have to cross the barrier of the epithelial cells. There's lots of lipids around. So how do they manage to get not absorbed at the epithelium, but then transfer back into the basal medium and to the cells?
Yeah, so in this case, you can see, because of the model that I've used initially for this preliminary model, you can tell that obviously from the concentration that we use, this is where we have to be kind of, use concentration that's been used in literature that's safe, but also you can't have it too low because then whatever gets uptaken, maybe there's almost none left for the neuroblast at the bottom to uptake. So which is why we tested a couple. And in this case, for levodopa, I guess because there's quite a bit remaining on top of the cell, this is where difficulty lays, where it can't really get uptaken by the neuroblast. But down the track, potentially, like right now, we're using the high performance, like HPLC. And if we're looking at low concentration, potentially LCMS for the samples would be used. And that may, improve the readings. And in terms of the future model, because they're meant to kind of both pass I guess you can say the neuron cells are a little bit more closer to the epithelial cells, then in the hopes that you'd have more uptake for the drugs before you actually reach the basolateral. So it'll pass through both the cells before we can check the transported.
So are you telling me you took a high dose in order to kind of saturate the epithelial uptake and then have the transport into the basal medium?
Not quite high dose. It's a dose that's been used in like a previous study before, but it's also why I've used 2 different concentrations to see whether, we can quantify the amount. And so far, like out of the 2, you can quantify the amount majority of the drug concentration. So the concentration that we use is good for now to test for further studies for the model. I hope that answers the question.
Not really.
Oh, sorry.
I still don't understand how you can penetrate the lipid barrier from the epithelium without being attached.
The transport, because from the epithelial, the drug gets transported to the basal. So obviously,
why is it not catch? It's a lipophilic drug. Why is it passing through the lipid bilayer of the cells?
The idea is, the drug is meant to, to be able to pass. We can't say that all of them pass through. That's why majority, for example, the very lipophilic drug, sertraline, majority does actually get retained within the epithelial cells, but we do have some that are transported through. And then if we look at, for example, levodopa, majority do stay on top of the apical chamber, but there are a little amount that passes through, get uptaken, and gets transported as well. Yeah.
There's another question down here. Towards the front. Oh, okay.
Yeah, thank you for your presentation. I was wondering, so you correlated with the log P, which is very often done, but what about the molecular weight? What are the 3 molecules having the similar molecular weight? So then you can just use the log P as a way to discriminate and then prove your hypothesis?
No, so the drugs we just chose like drugs that are brain targeting. In terms of the molecular size, they just happen to be quite, they're different, but not that different. At the end of the day, they're quite small. And that's why we looked into the charge as well. And in terms of the charge, that didn't really show, correlation as well as the lipophilicity and the log P values itself. Further down the track, we'll be, you can look at different drugs, like drugs that are bigger sizes with different, like a bigger difference of molecular sizes, etc., that are brain targeting as well for comparison. This is just a preliminary start, and these are the 3 drugs that we decided to work with for now.
And then there is an opinion that I would like to have from you. So I understood that the nose to brain effect is more agnostic to the drug, even to the molecular size. So you're able to really deliver molecules that would not necessarily cross the mucosa, the other mucosas, so efficiently. Do you agree on that, or you disagree on this?
I'm sorry, would you be able to repeat the second half? I think I missed it.
It's not an easy question. So there is the understanding that the nose to brain path would be agnostic to the type of API or even to the size of APIs. So this is a theory that needs to be maybe further proved. But do you believe on that, or because of the experiments that you have done with this cell line will just give you results that are similar if you tested other type of mucosas as compared to the actual nose to brain? Maybe it's a question that we can take outside.
Sorry, yeah, that would be great. Thank you.
Okay, I think we've got time for just one question. So if we could just go to the first one.
Thank you, yes. So first, excellent work. And what I'm going to say is totally unfair for a student at this stage. So you don't have to do all of this. I would highly recommend to do a fully factorial design on charge state, lipophilicity, and Zwitter ion, yes or no. Because that'll ultimately tell you what really the properties are. Of course, I understand why you did the project. You had to use existing drugs. It would be perfectly done in what you would call like an isogenic series of drugs. Yes. So yes, one comment for how much have you looked into whether the properties which you identify to be good for uptake happen also to be the properties which are good for effluxing from the brain after you reached it? Because you might optimize for the wrong thing if you don't consider both.
Okay, thank you for the questions. I think for that's, I guess, for future studies, will definitely be of interest to look into it further. Right now, it's very preliminary, so we haven't really looked that deeply into those aspects yet, but definitely down the track, that's definitely of interest. Yeah, thank you.
Thank you very much.
Thank you.